What is a cell? We've been talking about it a little bit already. The cell is the basic unit of life. No cell, no life. So this is a very, very important feature of biology, which is why we're spending so much time talking about it. A cell, as you know, is a membrane-bound compartment, which means that the interior of the cell can have a different concentration of pretty much whatever it needs compared to the outside. So you've got sort of a standalone area where different reactions can happen and different organelles can be housed.
Welcome to our prokaryotic cell. You also have a copy of a prokaryotic cell on page 70 in your book. This prokaryotic cell is prokaryotic because it has no nucleus. You are a eukaryote, you have a nucleus. So what we do with the DNA here is it's still in a chromosome, it's still in this long strand of DNA that's been wrapped around different proteins, but that strand of DNA is going to be circular in this instance, not linear, meaning that it does not have telomeres because it doesn't have an end. And that chromosome, because it's not locked inside the nucleus, is going to be floating around in the cytoplasm. Cytoplasm can also be called cytosol. So those are your two options. It's shown in highlighter yellow in this diagram. So this circular chromosome floats around right inside the cell. The area where it's floating around we call the nucleoid region. You can see right next to our circular chromosome something we call a plasmid, which is a small circular piece of DNA that encodes just a few genes. And what happens with a plasmid is that it stores some genes, and you can pass them over to another bacteria if you want to. But that's definitely floating around in the cytoplasm. You can also see these orange dots scattered throughout. Here, 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 all over the place. And those are ribosomes. Right? We have ribosomes as well. They play a key role in our central dogma of molecular biology. They're the ones that are going to translate RNA into protein. Now the ribosomes here have a little bit more freedom than what we would expect if our DNA was protected behind a nucleus. So these ribosomes can basically read RNA right away before it even falls off the DNA. To hold all the cytoplasm and the chromosome and the ribosomes together, we're going to have a membrane, right? That's a defining feature of a cell, is that it has this plasma membrane. And what you can see in the plasma membrane is that it's got two sides. Those are supposed to represent the bilayer. As you know, a lipid bilayer has a region with polar heads on one side, a region with polar heads on the other side. And hanging out in the interior are the nonpolar tails. And these nonpolar tails like to interact with one another. And so the way the bilayer is arranged is that you've got polar, nonpolar, polar, which means that polar molecules on the interior of the cell can't cross this bilayer to get out because they'd have to pass a nonpolar region. Similarly, if we have something outside the cell that's polar, it's going to be rejected by this nonpolar region. Whereas nonpolar substances won't even make it in there. They'll be stopped by this borderline, by the polar heads. So a membrane is sort of very well situated chemically in order to do what we need it to do, in order to keep the interior separate from the exterior. That said, we still need substances to come in and out. We still need water to come in, and if there's too much water, we need water to go out. If we are in the presence of food, we need to be able to bring that food into this cell. And so we need to have what we call a semi-permeable membrane, meaning that things can cross over. We don't want it to be too permeable, especially since we're dealing with a unicellular organism. Unicellular organisms need to be especially protected because they are their own last line of defense, right? As soon as something crosses into that cell, that's life or death for the entire organism. So in order to provide the extra layer of protection, we are going to have what we call a cell wall, which you can see in blue. So on the inside, we have this cytoplasm. then you've got the lipid bilayer, then you have the cell wall. Bacterial cell walls need to be especially strong. They need to be especially strong, and that is because, as I said before, it's the last line of defense for the entire organism. So not only is it providing structural support, not only is it sort of limiting what can get in and what can get out of the cell, but it's also going to be protection from things that might want to attack this bacteria or whatever other kind of prokaryote we're dealing with. All right? One such thing could be enzymes that you produce, right? That's why we make lysozyme in our snot. That's why eggs make lysozyme in the egg whites. It's so that, you can try and fight off bacteria. Because if you can get through that cell wall, the chances of killing that cell are much higher, right? It's a very strong protective layer. So if you can interfere with it, that cell has, trouble on the horizon. All right. Now, with that said, let's move to some of these other exterior features. You can see in pink what I call the capsule. Not every prokaryote has one of these. Some of them do, some of them don't. But basically what it is like a jelly-like coating around the exterior of the cell. It's sort of another protective layer to have around the cell. We also have flagella, which you can see here. They look like tails. And these flagella are going to be our little motors to help these cells swim around. And you can see a link down below in the description area so that you can actually see some cells use flagella as a means of locomotion. If you're a single-celled organism, you've got to take care of everything in that single cell. And the fact that you can get away from danger and move yourself towards food, that's going to be a big point in your favor if you're thinking about life and death for a single cell. To be fair, locomotion is also a big deal for us multicellular creatures. But it is kind of amazing how much these guys can move around and how much they can maneuver using these sort of simple projections. When we talk about prokaryotes and bacteria specifically, towards the late winter, early spring, we'll come back to some of these other structures. But the main ones that we really care about, we've got our circular chromosome, we've got our plasmid, we've got our cytoplasm, our cytosol, we've got ribosomes, we've got our cell wall, we've got our plasma membrane, we've got our capsule, and I think we said flagella. So that's the prokaryote.